Oh, we've closed the doors? Okay, good. So welcome back to the last session of today. It's my job to introduce Professor Sloot. But before I start with the introduction, I'd like to read a quote about computer science, which I like. And it's actually often misattributed to a Dutchman also, Dijkstra. And the quote goes something like this. Computer science is no more about computers than astronomy is about telescopes, biology is about microscopes, or chemistry is about beakers and test tubes. Science is not about tools, it is about how we use them and what we find out when we do. So this was a cry to people in the US, I think in the 80s, to understand what computer science actually was. The interesting thing is now, if you look back at that quote, computers are no longer the tools of computer science, but the tools of science. And that's where the field of computational science really starts to come in. And it was really, Peter was one of the first worldwide who started to recognize the field of computational science. So this idea that it's not really the computers, but it's the methods, it's things like algorithms, it's things like models, which are really the important things. And I think this applies also to complexity, very much so. I think computational methods are very much a part of complexity science. So given that he's a professor of computational science in Amsterdam, he does this with a background in chemistry. He has a master's in chemistry, he has a master's in physics, and he has a PhD in what I will say is computational science. Don't call him a computer scientist, because he gets very upset by that. I learned that. So he spends most of his time, well, some of his time in Amsterdam. He also was awarded the Leading Scientist Award in St. Petersburg, Russia, in 2010, which is actually the largest individual award in science worldwide. Luckily, we have him in NTU also for 4 months a year, so I get to spend some time with him while he's here. And what can I say? I've known Peter very well, actually, for the last 3 years. So I know his dislikes, for example, don't call him a computer scientist. But I also know his likes, and I know what really gets him excited. So if you do want to animate him, if you want to get him excited, then you just simply talk about complexity. Anytime you do this, he will talk to you for hours and hours, even if you don't want that. But it's always, for me, it's always very, very inspiring to listen to him, because he shares the same views as I do, but he also has the background knowledge that many people don't. And he also has a highly creative mind. So he's one of the people that will sit and talk to you and come up with crazy ideas and really spend time kind of fleshing out these ideas. I'm sure many of you have experienced Peter's kind of interactions at NTU, and they're always insightful, they're always very engaging. And I'm sure today's talk will be no exception. So with that, I'd like you to join me in welcoming Professor Sloot.
Thank you, Mike. Okay, so I came up with this title. We've been doing a lot of work on modeling. So I think Murray Gell-Mann was talking about world modelers, and I said, yeah, I think I feel like being a world modeler. And so I came up with this title because we did quite some work on looking at the way information is being transferred from a virus point of view. Now, there are two ways I can tell this story. You can really sit inside the virus and look from the virus point of view, or I can take it from, let's say, an epidemiological point of view. What I'm trying to do is I will try to do both, just to give you a feeling that when you look at things slightly different, you might eventually come up with good answers. So this is the world clock, the one that I like to look at every now and then. This gives you kind of a feeling of where we stand. This is counting since the beginning of this year. And so we have a world population that is about, well, just past 7 billion, as you know. And so what you see here are a couple of interesting numbers. I'd like to point you at the diseases here. So from tuberculosis to communicable diseases to hepatitis, malaria. So you see they're really huge numbers. They really account for a serious amount of death that we have in the world. There are other interesting numbers here, and it's still counting, of course. As being a Dutchman, I'm also very much interested in the amount of bicycles produced. Actually, you see the number comes very close to the population growth. So for every new kid, we have a bike. So that's good. But the message of this slide is we are with a lot of people, there's a lot of stuff happening, and a lot of the stuff that is happening is because we interact. And that, of course, results in problems. And these were laid out these are laid out in this graph, where we look at the relatively likelihood of some kind of disaster to happen against the impact that we expect that it will have on the world. And so there are things like severe weather. Well, we have seen some. But look, the one thing that really stands out is this pandemic influenza. There's a very high likelihood that we're going to be hit by yet another pandemic, a pandemic disease, I should say, not only influenza. And the impact will be very strong. But we have had it, through the years. We have had, of course, the pest in Europe. We have had this huge influenza in the 1900s in Europe. We had tremendous amount of comparable disasters, I would say, that really hit us hard. And there is a pandemic that's going on, and that actually urged the European Union to ask this question. They put it out in the wild, so to say. They said, if we would have like €1 billion that we could spend on stopping the pandemic HIV/AIDS, what should we spend it upon? Should we spend it on getting better medicine, or should we spend it on changing behavior? And these are the kind of questions I like, because when you start to think about that question, within a few seconds you realize, when we talk about better medicine, we talk about molecules, right? And we talk about chemistry, and we talk about immune system and toxicity and whatever you have. We talk about changing lifestyle, changing behavior. We talk about sociology, policy, money, and psychology. And so the question is, if you look at this, you see how can we ever find an answer to a question like this? It's it's really comparing apples and pears and cows and whatever you have. So in order to get an answer to that, you really need to have a crude look at the whole. And then you have to have a detailed look at the parts. And then you have to find a good look at all the holes that you have missed, right? So I'm going to have a crude look, a detailed look, and a good look at the holes. Okay, so this is the situation. This is the HIV pandemic, as it stands. As it stands basically, as it's recorded up to 2010. And so the dark areas are, of course, the places where the highest incidence is. And we see that the amount of people infected with HIV is about 14 million people in the world. Now, this is a dramatic underestimate. This is what we really know that is true, but it's probably much higher. The reason why it's not higher is, you already guessed that, of course, is because many people do not know that they are infected, right? And there are many areas where we don't have correct samples. So this is a very strong underestimate. Now look at this in reflection of the first slide that I showed you, that we are 7 billion people. We're very much connected. We're flying from one place of the world to another place of the world. So everything is connected to everything, and everyone is connected to everything. So these kind of pandemics are very, very hard to stop, and very, very hard to really assess and to understand. So where does it take place? This is the world, and you might recognize it. And this is happening if you scale each country with the amount of HIV that's in the country. So here you see, this is where the problem is, and this is where the problem is. And if you now would scale every country with the amount of money that's going into HIV research, the world would look like that, right? So the money is going where the problem is not, right? And so I think it's our task to really make sense of that amount of money that we're spending, to see if we can get a good answer to those questions and to the problems that are there. Now, HIV is tremendously complex, and I'm going to take you through it in a couple of minutes. And I'll be very concise about it, but I need to bring a couple of things across. And like the previous speaker, who at the beginning of her talk said, at the end of my talk there will be a twilight zone. So that really keeps you on the edge of your chair, which is a very good trick. I'm going to do the same trick. At the end of my talk, I'll be talking about sex. Okay, but before we get there, So this is basically the problem. There are many aspects here. Just take you very briefly to it. This is the artist impression of the HIV virus. We have all kinds of drugs. We have about 24 drugs. So I'll say a bit more about it later. There's drug resistance. That means that the virus won't work anymore at some point in time. There is an immune response, which we hardly understand. There is an infection process in which we probably understand only 40%. There's no understanding at all at this point in time about toxicity and degradation, and the inhibition effect is also quite poorly understood. So this is the problem, and this is where we have to really get an understanding. Apart from that, there is the problem of the spreading of the virus through the population. So yeah, it's a truly complex problem. And the only way to address that is, like Neil Ferguson said, he's a very famous epidemiologist. He said the only way to actually look at these kinds of problems is to take a complete holistic view. You cannot look at these things in isolation. You have to look, well, of course you have to look at them in isolation, but at the same time try to make the connections to all the different parts that constitute that system that you want to understand. Which is, I think, more or less the definition of a complex system. So that's what we're going to do. And size matters in that case. If you look at the scale of things that you want to address, if you want to have a bit of an understanding of what's happening, you really go along many scales. Murray Kalman was quoted by John Hollands saying that you get a complete new science. What was it? Every 10 to the power. Maybe you said that. I forgot. It was like every three orders of magnitude. And so look at this. So we're talking about processes that take place on a scale of 10 to the power minus 10 meter up to something like a meter, right? Or 2 meters if you like. And time scales 10 to the power minus 14 seconds on the DNA replication level, all the way up to a lifetime. So there's not one science. There's not one science that can cope with this. We cannot, design an equation here, right? And then just track that thing all the way through there. There's no way. So we have to think of a more integrative way to deal with this. Now, I do believe in systems studies and system dynamics, if you like, but I don't think that system dynamics makes sense here, because there will be no covering equations or something like that. You really have to, break it down in bits and pieces, try to understand these bits and pieces, and then see if you can find mechanisms to glue them together in a sensible way that you can actually validate. And that part of validation is crucial, which is quite often skipped. So what I'll do is I'll take you through the essential parts so that you can hopefully appreciate the complexity. The virus, again, this is an artist impression, and once an individual gets infected, the individual produces about 10 to the power 9 new viral particles per day. That's quite a lot. So per day, 10 to the power 9 new viral particles. Now, lots of those are not fit in the sense that they will actually be eradicated by the immune system, but lots of them are. So, and then what happens is something very special that I really want to draw your attention to. HIV is an RNA virus. It's a retrovirus. It doesn't contain DNA, but it has RNA in it. And a virus is a dead thing. It's only becoming alive, if you like, because of us. Right? So what it does, it hijacks the reproducing mechanism that we have in our cells. It hijacks the fabric, if you like, the machinery in your cells to be replicated. Now, our cells work on DNA. They don't work on RNA. So what it needs to do, it needs to transcribe the RNA, which is in the virus, to DNA. Now, that transcription has been done by a molecule which is called reverse transcriptase. And that's a kind of stupid molecule in the sense that it makes mistakes. So while it's reading RNA to produce DNA, which then will be inserted in the cell, it's making mistakes. And these mistakes are basically the mutations that cause the the virus to change. We'll get back to that later. But that's the biggest problem. That's really the biggest problem. So once you design a medication, because of this replication error, the medication might not work anymore. I'll show you. So what we want to do is to basically track this whole problem of HIV from molecule to man, as I like to say. And we're going to do that in 4 steps. And I won't take you to all the steps in detail, but just give you a flavor of how we do this kind of research and what we have obtained so far, and some hot new results that we have. So we were doing something like molecular simulation, something like agent-based simulation, something like cellular automata, and something like complex networks. And then somehow we have to make sense of all these different things in a good way. So here is a, let me see if it works, yeah. So here's a kind of an animation of the viral process. So the green thing is the virus. This is your white blood cells, your T cells, the T lymphocytes. And you see different steps and different drugs. An inhibitor is a drug, a medication. And we do have medications that actually block the first attachment of the virus to the cell. But these medications are not very good and not very often being used. If that fails, then there's a process going on which is called co-receptor binding that actually makes the virus more strongly connected to the cell. If that process is not blocked, then we get the fusion, which is actually a kind of melting, if you like, of the virus membrane with the membrane of the white blood cell. And then what happens is that the virus actually, it's like a programmer. It's now just giving its code to your cells to be replicated. Here is the RNA. There are all kinds of supporting molecules, like, for instance, the reverse transcriptase, the molecule that I mentioned, that needs to do the transcription. And again, we have blockers there, and this is the important blocker that's being used mostly nowadays, is reverse transcriptase that tries to block that process of transcribing from RNA into DNA, and where all these mistakes are being made. Now, then, if that blocker doesn't work, then the virus is actually entering its viral DNA now into the DNA of your white blood cell, which then happily starts to copy because, that's the thing that cells do. They want to copy, and by copying themselves, they also copy this information, this program. They run this program of the viral and actually produce new virus particles. And that's happening. There are new virus particles, but they need to be compartmentalized. Think of the story of John Holland about boundaries. It needs to get the boundary to actually do something. The animation is doing, it's still working. So it needs to get the boundary, and that's what's actually happening. It's going to aggregate. And once it's aggregated, it created this polypeptide boundary, and then it actually releases again. And that with a production rate of 10 to the power 9 viral particles per day per patient after primary infection. Okay, so you got the idea. Now the problem is these mutations, right? And well, we know if we have a drug that is working, and then for a certain virus that's in the body, and then these mutations occur, then after 2 mutations, let's say 2 fit mutations, we already have a 4-fold resistance to the drugs. So the drug is just working only, for 1/4 of its power, so to say. With 3 mutations, already a 10-fold resistance. And like I said, there will be many mutations. So the resistance is the biggest problem of HIV. And understanding the transmission of HIV and understanding this resistance and understanding the transmission of resistance in the population is the hottest topic in this area of science. And here's the situation. We have about 250,000 people in the US and Europe living with drug-resistant viruses. And 10% of these viruses, of these new infections, are with drug-resistant, so 10% of new infections are with drug-resistant viruses. And a small portion of these patients die because we just don't have any methods, any cocktails that we can think of that work because they are resistant to all of them. I can talk about this forever, I won't, don't worry. But this is a really interesting topic, to really get a good understanding of that. Now, in order to come to this question of the European Union, we need somehow to get from this molecular description all the way up to this description here. So I'm going to do that in a couple of steps. And the first is on this medical stoppage step, if you like, this drug step. So here what you have is a reverse transcriptase, again, that famous molecule that transcribes from RNA to DNA. And here is the drug, this is the medication. So the way these things work is, you probably remember that from high school. The way these things work is the reverse transcriptase tries to read this part of the RNA and then produces its DNA, right? But it reads with a small part of the whole molecule. So a small part of this whole thing, this whole spaghetti string, is actually the active part. And that's the part that's there where the yellow, what is it, green-yellow thing resides. Now, if you would kind of block that part, if you would put a cork in it, which is this yellow thing, if you would just block it, then it cannot read anymore, right? So that's the whole way these medications work. You just block the active site of the molecule that is the culprit here. So what you do is you design a molecule that fits perfectly in that bigger reverse transcriptase molecule. If you do that, you know, you're in business, if you can manage to do that. And so we've been studying that in quite some detail together with the group from Peter Coveney in UCL. And here's an example of the many simulations that we've done. So here again is the reverse transcriptase. Here is the molecule that we insert on the active site. Here the red spot is the active site. And the thing that you see here is the thing that you do not want, right? Because what you want to put the cork in place and it should stay there. But what's happening, and I'll run the thing again, if I manage to do that. So what's happening is we put it, you know, in the computer, we put it in place, but then it walks away. And the reason why it walks away is because of the mutations. So there are mutations here, there's a mutation there, you don't see that because that's too small. But because of the mutations, you change the spherical interactions and the spherical hindering processes in that big molecule, and then basically the medication walks away. Now, the only thing that you need to remember up to this point is one thing, that we can actually calculate the binding affinity of this medication of this reverse transcriptase. So we can calculate how likely it is that the medication will work, okay? That I call the binding affinity. If the binding affinity is 100%, that means the medication works 100%. But it's not 1 or 0, it's something in between, right? So it can be 30%, 20%, whatever. Now, this binding affinity is important because if the binding affinity is very high, right, that means that the viral load is very low. That means that the probability of transmission will be very small. So there's a direct link. Here you already see how simple actually this work is, because you really see already the link between sexual interaction, the transmission of the virus, and that molecule, right? So if the binding affinity goes down, the viral load goes up, and the transmission probability goes up. So we will make use of that later. This is the thing that helps me to connect models together, right? Here I have basically solving Newton's equation, and here I will be solving something else. You got this idea? Good. So, but that was a simulation, right? So the point about simulation, I like to give you this quote. Who recognizes this guy? If Murray would be here, he would probably know. This is Wigner, very famous physicist and mathematical physicist, I would say, who introduced, and then he said, after watching a successful computer simulation, I think the computer understands it. Now I want to understand it, right? And this is, of course, the trick that we have to be very much aware of that. By the time we were doing all these beautiful simulations, you know, we still have to be sure that we understand what we're doing. But then on the other side, the whole thing, most of the complexity science studies are with computers. You know, we are building models because this is the only conceptual framework that we actually have. So we need to do that, and we need to always go back to this point, you know, do we really understand what's happening here? Well, I won't go into the details, but what we did, for instance, for this specific case of calculating this binding affinity, we did actually do laboratory experiments in which we calculated the amount of free energy, and we compared that with the simulations, and that was pretty well. As you can see, the numbers are very close. And this is for different mutations in the reverse transcriptase. So we can actually do it. So having solved that, we have a binding affinity. Now we come to the real hard part. Well, one of the real hard parts. And that's a part where we have to take into account the immune system. Now, John Holland here said, the most complex system I know of is the immune system. And I think I agree with that. But even then he said, even though it's the most complex problem, with one vaccine, we can already act on it, right? So even if it's complex, very complex, there might be some simple principles underneath it that we might want to, that we can make use of, which is what we did. And so what we actually did, I won't go into the details, just give you the result. What we actually did is we made a cellular automaton model, a three-dimensional cellular automaton. This is a two-dimensional introduction to that. A three-dimensional cellular automaton model in which we just said, okay, if the cell is here, if a virus is here, what is the probability that it can actually be depleted by the virus, by the cell? What is the interaction between the cells, etc.? So there are there's generic principles that you can do. And then you can just run to all kinds of simulations by looking at how virus particles will be depleted by the cellular system. And you work through that, you work through that. And if you do that, and then after a couple of years, because it took us really 4 years to do this, you are actually able to reproduce the dynamics of the virus. And this is a slide, the second point that you want to remember from this talk. So what you have here is a patient, and what you have here is our simulation. And what we're looking at is the two things. The black dots is the amount of virus particles that's in the body, right? And the white squares is the CD4. That's a molecule that sits on the T cell, and let's say that it represents the health of your immune system. So the higher the white square is, the better it is. Now what happens, and these are weeks, and here we go to years. What happens is the following. After the primary infection, so an individual gets infected with HIV, then this explosion, real explosion of viruses. Remember, 10 to the power 9 per day. And then the immune system starts to kick in, and it actually starts to basically reduce the amount of viruses, but at the cost of something. So the immune system collapses, and you don't feel that well at that point in time. Then the immune system recovers, and the virus seems to go down. If I would stop the picture here, if I would stop the slide at this point here, you would have basically influenza. This is the way our immune system works, right? It just depletes the viruses, and it recovers. But the point with HIV is it doesn't recover. And the point is it doesn't deplete the virus completely. I'm not going to tell you why, but that's what's happening. And then what happens is that the virus actually starts to hide in the lymph nodes. And it really hides. One of the ways it hides is actually it kicks away some molecules on its surface, so the immune system cannot see it anymore. The immune system becomes completely blind for the virus. It's an amazing trick. And then the virus slowly starts to increase again, and now we're talking about years suddenly. And in the meanwhile, this is called the clinical latency phase. In the meanwhile, the immune system starts to collapse, right? And then we can come into this. So this was the primary infection phase. This is the clinical phase, and this is then the AIDS phase. That's the moment where the amount of viruses is so large and the immune system is so much compromised that you get all kinds of collateral diseases coming up, right? So if you then have a cold, it might actually kill you because there's no immune system to recover. But also, you know, our immune system protects us from cancer. And so a lot of cancers actually come up here in the moment that the immune system is collapsing. Okay, so the second thing, so the first thing you remember from my talk so far is this binding affinity. The second thing is this model of this is not a model, this is a real patient, and this is the model, but you see it's a very good comparison. The second thing you remember is this, you know, this peak going down, hiding up, going up again, and this collapse of the immune system.
Has the virus changed a great deal by the time?
Excuse me?
When the virus recovers, is it a different virus then?
Yeah. There are all these mutations, and it goes through this fitness landscape, and you have actually populations of viruses. Yeah.
Can you say why the immune system suffers during the latency?
Well, the immune system suffers during the because that's a good point. So even though that's a good point. So even though the virus becomes mostly invisible for the immune system, that doesn't become completely invisible for the immune system, and you get all kinds of what you call breakdown products that will actually trigger the immune system, and it cannot actually handle that. Yeah, very good question. Okay, so with these two things, we actually now have something that we can start to think about. Okay, that's all happening in the body, but now we have to take the thing outside of the body. So then we need to talk about sex, like I promised. And because of our study is done in different cities, I'll talk about sex in the city, and So I will be talking about how we take information from social information, behavioral information, medical information, doctor records, etc., and how we use that to actually combine with this model. And we do that for different cities, for Amsterdam, Rome, and San Francisco, because that's where the data is. And okay, so we have to include, now we have to start thinking about, okay, what's happening there? So what I'm going to do is I'm going to digress now. I'm going to digress and tell you about why we choose this specific model to model the transmission of viruses. Because what complexity, for me, complex systems research is about, and where I spend most of my time, is thinking, okay, I got this complex system, this is the data, what is my model? And where does my model sit, right? If I have a model which is too highly granular, there's too much details there, I don't have data, then it doesn't make sense. So I have to kind of fit my model with the data that I have. And then again, I need to make the model such that I can hook it up with the other models that I have. Because if I have a beautiful model of transmission, and there are beautiful models well known in epidemics, and I cannot combine that with, for instance, treatment models, or I cannot combine it with behavior, or I cannot combine it with the binding affinity, I can just throw it away. It doesn't make sense for me. So I need to look for models that actually fit this sequence of models, if you like. So a way to study that, as I'm trying to convince you, will try to convince you now, is making use of a thing which is called complex networks. Now this is a complex network. This has got nothing to do with sexual transmission. This is a dating diagram, or maybe it has got something to do with it. This is a dating diagram. So this was interviews done at a high school in the United States. And the blue ones are the boys, and the red ones are the girls. And the interview was about whom are you going to date the coming 2 or 3 weeks, right? So from that interview, you can actually find connections. And that's the message you get from here. So the message you get from here that this boy was dating 2 girls within a certain period of time. Now you can ask yourself, there are some very happy boys here. And pretty active girls also. I forgot where, but there are some pretty active. And you can ask yourself where you yourself were. I think I was around here. But you got the message, right? You got the message. And the message is that we can actually make this kind of graphs to express 2 things, individuals and the interaction between individuals. And that's exactly what I want. Because up to this moment, I was talking about what's happening in an individual. And what I need to start talking about is what's happening between the individuals, right? So this is actually a nice way to go from the virological description to the epidemiological description and connect them, which is, again, the crucial question. How do we connect all these models? Because I don't believe in a panacea. I don't believe in one model that does it all. I have to connect models. These things can get very hairy. I skipped it one. And I have to say a few more things about these networks in order for you to appreciate what we can do with that and how we can make them actually work for us. You all heard about small world networks. The story is there that we are all connected, right? So if I want to basically send a message to anybody, anywhere in the world, I might know you, and you might know him, and he might know, and then within 6 steps, we can cross the whole world. That's the message. So that's, if you like, a small world vision of the world. So even if we're talking about 7 billion people, we're only 6 handshakes away, right? That's more or less proven. I'm careful here, but let's say that it's proven. So that means, in my case, that's me. I was an opportunity at some point in time to shake hands with this guy. This guy shook hands with that guy. So that means that I'm 2 handshakes away from Barack Obama. So after this lecture, feel free to shake my hand. And that gives you the idea. Now the point is that we always thought that that was the way these networks worked. So we got this idea, the K stands for the amount of connections an individual has, right? So we always thought it would be like this, or there will be, as everything you were saying, that also most things that we look into in the world are somehow normal distributed. So this is what we thought it would be. The probability of having K connections would be something like this. There would be, the majority would have, let's say, 10 connections, and a few would have 100, and a few would have 1. But unfortunately, or maybe fortunately, if we look into real-world connections, they don't behave like that, they behave like this. And this is a scale-free. This shows that the system is scale-free. We call this scale-free complex networks. And what it means is that the ones with, let's say, few connections, there are most of them, and the ones with a lot of connections, there are few of them. And this behavior you will see throughout all kinds of networks, all kinds of real networks. Whether it's networks that come from, this is the plane connections, the flight connections in the US, or it comes from science co-authorships, something completely different. Again, they have this scale-free structure. Or if we look at another one, we look at the structure of organizations, which is an emerging property, right? So it seems that the emerging property of these kind of interactions are always scale-free. But also, and you can do things with it. Once you have an understanding of those networks, you can actually see how, for instance, opinion spreads through it. So this is research that was done by Xi et al., who actually looked in the way opinion spreads through those networks. That's good. Now we have a model. We can play games with that. This is work we did ourselves on protein networks. So you can really map out the protein networks and then again see that these things are scale-free, having this log-log behavior. So let me say a few more words about this, again for getting a better understanding. These models, so these networks can be modeled like this. This is the reason why they're called scale-free, because if I change k for ak, I basically get the same function. So this says the probability of having k connections is k to some kind of power, right? So this means, and here I need your attention, this means that with one parameter, gamma, I can describe that network. So if the network is characterized by this. So with one parameter basically describes the interaction between us. I think that's nice, especially from a modeling point of view. So, but as John also mentioned, these networks are not static. They are dynamic. They are growing. They are rewiring, etc. And so, just a way for you to start thinking about how these things look, what they look like. This is, of course, a toy model, but this just gives you the feeling of how these things actually emerge. So this is the size of these balls tell you something about how many connections they have, right? And the more connections they have, the higher the likelihood that they get more connections. So the rich gets richer and the poor stay poor. That's the idea. So this we call the hubs, and the other ones are, let's say, the peripheral nodes. And this is typically the way these networks emerge. So it's like I said, the rich gets richer model. It's also called preferential attachment. Now, if you remember high school mathematics, you will see that if you integrate this thing to calculate the average, that thing will diverge for a value about gamma is 2. And if you look at the variance, so the spread in this function, you will see it will diverge for a value of 3. So that means there's lots of strange dynamics happening there. It also means, I'm not sure if I can say this, it also means that because the average actually doesn't exist at these values, that means that all our learning algorithms will not work on this. Because learning algorithms expect an average somewhere. How can you else calculate a cost function? So this makes these things really incredibly difficult to study. Okay, so these things are scale-free. Now the point is, if I have a thing like that, and now I would like to disrupt that network, right? And that could be a criminal network, but it can also be a sexual network, or it could be any other network. If I want to disrupt it, what I can do is basically, start shooting at it. So that's what I'm doing here. So now I'm starting to shoot, and this is a random shot. So I just randomly shoot at these balls, and what you see is 7 nodes. And the whole thing is more or less is still intact, right? I can still get information from here all the way up to there. It's still somehow connected. The network is still, the integrity is still preserved, if you like, even after 21 removals. Now, that is if I do a random attack to that network. If, on the other hand, I do a targeted attack to that network, So that means that I start to hit the richest ones, if you like. So I'm going to kill those that have the highest connections. And already after 7 nodes, killing 7 nodes, you see the whole thing basically falling apart. So here you see the importance of understanding the structure of those networks in terms of controlling those networks or changing their behavior or changing the way information goes through it. So that's the message that you want to keep here, that you want to take home here, is that the structure of these networks is pretty easy. And that if it's like that, we might be able to, first of all, model them, because we can easily generate networks like that in the computer. That's a simple thing. And we can actually interact with them. Now, there's another characteristic which is really very new. It's a thing that we discovered ourselves, and it was published 2 weeks ago or so. That is that in these networks, if you apply noise to it, then it seems that they are better to propagate information. Now, listen what I'm doing here. I'm talking about information. Well, the beginning of my talk was about viruses, right? So this is the way I look at things. So, you know, a virus is just a bit of information that you want to propagate. Or an opinion is a bit of information. So information is the fundamental unit here. Now, what you're looking at here is, again, an artist's impression of something that is more deep. And it tries to give an answer to the question, what happens if I have a network that I just showed you, and I put information here in that network, and I measure how much of that information appears at the end, some end of the network, right? I measure that, and then what I do is I superimpose errors. So instead of making the shortest path, you know, there's just a rewiring going on that I have to go like that, okay? So by introducing errors in that way I send that virus or that opinion or whatever you have, that piece of information from A to B, by introducing errors, which is noise, I do that on a temporal situation, then actually what's going to happen, that the network which is somehow in some kind of optimum can actually then just move to another optimum. But the point here is that I have to drive that noise somehow in coherence with the dynamics that's already in the network. That's why this thing is called stochastic resonance. It's a resonance effect because of the stochasticity in the system. This, of course, is a 2-dimensional thing, but the networks that we were studying are highly dimensional. And the thing is that you have to drive it in such a way that this switch that you see actually synchronizes with the amount of uncertainty that you have in the system. So we did that for many systems, and one of the things where we did it for was an email network. You're doing something. So what we did here is this is a very famous email network that's publicly available. It's from a company, Enron. It was a company. And maybe that's the reason. Because they shared their email network. And the question you can ask yourself, okay, what would happen if I sent information from this part to that part, and then I disrupt that network slightly and see if I can still get that information from one individual in that network, from one individual in that company to another individual in that company. And we played around with that, and we found that it actually goes through an optimum. So what you see here, it goes through an optimum, and what you see here, it goes through an optimum. And this is the red spots are the optimum. So what I would expect when we started to do this work, I thought, you know, introducing noise will make it harder to get information from one side. But what happens, by introducing a little bit of noise, it will make it easier. So noise helps you to get information from one side of the network to another side of the network. I think this is a fundamental aspect of complex systems. But this is, let me finish this part, because it's a crucial part. This is just a conjecture, right, this remark. It might well be, and if you put too much noise in it, then actually the whole thing doesn't transmit anything anymore, right? So no noise, you have a certain efficiency. Too much noise, you have a low efficiency. And in between, you get a better efficiency. That's basically happening. And so it seems that noise is a crucial thing. It might be, and this is again a conjecture, we didn't study that yet, but this is what I'm doing now, is it might be that the reason why these networks are the way they are is because they have this aspect. It might be that they are in some kind of phase space that they constantly need to be able to adapt to noise, to changes, to errors. And so we're doing our work on calculating this for protein-protein networks. Your question? Okay. Yeah, sure. Both. So it's exactly these two. It's exactly the way we select noise and the ways the things are being linked. And these are these P and Q. These are the two different things. Yeah. Okay, good. So, but this just gives you the flavor. I was talking about characteristics of those complex networks, right? So this is another new characteristic that I would like to give to you because it's a nice result, I think. Now, this whole research on complex networks is really, really booming. And it's one of these paradigms that we want to use to study complex systems. And chaos was another one, right? And catastrophe theory was yet another one. And what else we had? Your neural networks, fractal description, self-organized criticality, the pair buck stuff. These were all paradigms which suddenly popped up and were being taken over by all kinds of researchers in the field of complex systems. Whether you were talking about ecosystems, you were talking about the brain, or you were talking about social cultural systems, these kind of conceptual frameworks were very rich and very much used. And what you see here is the kind of publications that these different theories and methodologies, if you like, produced. And if I would superimpose into that the publications that were up to 2010 for network research, so you see that's completely exploding. And the reason why it's completely exploding is because it's such a rich concept. It's such a tremendously rich concept. You can model opinion spreading. You can model obesity spreading. You can model, well, whatever. So I think that's really one of the reasons. I'm not saying that it's a panacea, but it's something very useful. And we're going to use it. So the steps ahead. Right, what I now need to do is now I need to link all the story that I was telling before, what's happening in the body, with this network story somehow to make sense of it. And so what I need to do is I need to infer the sexual networks from the data that I have. I need to, describe the nodes in the networks and the links in the networks. And somehow I need to take into account all the data that I can get my hands on. Right? There is no shortcuts here. I really need to get as much as I can to make sense of it. So I need genetic information, medical behavior information, whatever you have. And then hopefully have a mechanism to glue that together. And then, of course, it's crucial, making models doesn't make sense if you cannot validate it. So it's crucial to somehow validate it. And then if you can validate it, you only hope, it's a hope, that it makes sense. If you can validate your models, that you say, okay, it explains the historical data, then maybe it's also predictive in the sense of, telling you something about the future. That's a maybe there, of course. And then the third question is, okay, once you know all that, things that decision makers would like to know, that medical doctors would like to know, that the government would like to know, is can we actually control it then? So if we know how this spreading is taking place, can we control it? And my first reaction when we started to do these things was, of course we can control it. Once we have it, we just play around with the parameters, we can control it. Well, that's not true. That might not be true. Let me put it like that. It actually reflects on the remark I made yesterday, that there is a certain arrogance we have, that by the moment we think we understand how to describe a complex system, we also think that we can control it. But that might not be. Any questions up there now? Yes? I mean control, in the sense of changing things in the way I want it to change. Yeah.
Not influence?
But no, influence I can always, because I can always check out an individual, or I can do something. But control means that you influence in such a way that it goes in the direction you want it to go. That's the question. I'm not saying that I can.
Oh, okay. Oh.
There was a question mark here.
Okay, I feel much better.
Let's wait until the end of my talk. Okay, but these are the kind of questions we want to ask, right? So how do we infer these networks? How do we validate them? And if we kind of, if it makes sense what we're doing, can we control them? So here we have things that also are called ridiculograms, but basically networks. This is actually, we went to the Center of Disease Control in Atlanta, and we dug into their data. And we came up with the heterosexual and the homosexual. This is men having sex with men. That's the formal terminology people are using, MSM. So from now on, I will use MSM because it's a better denominator. So the MSM and the heterosexual population. So you can actually dig into the data there. I'm not going to tell you how to do that all. But the bottom line is that we found that for the heterosexual population of the United States, the gamma is about 2.7. So again, remember, the equation that describes this function says it's the probability of having K connections is K to the power gamma, and gamma is 2.7 for heterosexuals, and it's 1.6 for homosexuals. And the K max is about 70, so that means this is about the amount of sexual contacts the maximum amount of sexual contacts per year. And that can go up to 250. Different sexual contacts, I have to be careful here. So with different individuals, the amount of different sexual contacts per year, up to a maximum of 250, typically in MSM in the States, and about 70 for heterosexuals. That is something you can talk over coffee. Not now. But the thing there is that with two parameters, I characterize those dendrograms, right? I characterize those graphs to some extent. Of course, this is simplification, we all know. And it's a snapshot, we all know. And things are changing over time, we all know. But it's the first good approximation. Now, the thing that you can do is actually add to that information that you have on social interactions. Or things that, so on average, we know that in heterosexual populations, the connection between the two individuals is actually less when the distance in age gets larger. To say it differently, people prefer people for that the age difference is not bigger than, let's say, 40 years. And so you can actually filter out those that, if you look at the total population, you can filter out those that do not comply with these kinds of social rules or with some probability. So you can do that. And if you do that, then you can actually get a good idea of how these networks evolve. So here we start out with everybody is connected to everybody, and then we just apply those kind of filters, social filters, behavioral filters. And there's also sexual preferences filters, of course. And then you sort it out, in this case, in 3 populations. And this is the population heterosexuals. This is the MSM population. This is the MSM population. And this is the drug users population. These are populations all infected with HIV. So it's not the total population, it's the HIV infected, because we have patient data there. So we have all this additional demographic information. Okay, so that's what you can do, which is pretty useful, because then you can start doing simulations and analysis of that. And remember, I was trying to get all kinds of medical and behavioral parameters out of the system so that I can plug it back into my models there. So here you see this is the time of seroconversion, meaning the time that people got infected. And that's for the drug users, for the homosexual population, and for the heterosexual population. So the drug users were much earlier in time already infected with HIV. So there was really a peak that was before the peak of the homosexuals and the heterosexuals. So these are the seroconversions. This is all individuals. This is real data, real patients, real connections. And so that gives us some confidence that we can do that. And then what we did last year, or actually it took us a couple of more years, but last year we published it, is you can actually combine this social network, if you like, because mostly this is social information, right? What kind of sexual preferences, what are the ages, et cetera, et cetera. And you combine it with genetic information, because we have the phylogeny, so that's the genetic information of the virus per individual. We have that also. And that's this information. And so we can actually combine these two to get a much more clearer estimation of these, this famous scale-free function. So you get a we fine-tune, if you like, we fine-tune this whole dendrogram, this radiculogram, we fine-tune that with genetic information and social information. And this is for the homosexual population that we studied there. So again, that gives us a number which we can then use in our simulations. So now we can bring things together. And so what we can do now is ask ourselves, okay, remember, binding affinity, that's one thing, which tells me something about the success of applying a certain therapy. Immune system response, which tells me something about how much of the virus will be depleted, recovered, etc. Also where the patient is, because if the patient is here, and if, this is an important thing, if the patient is here, let's say in week 5, then if there is a sexual contact, an unprotected sexual contact, then the probability will be very high, right? Because you have this high number of virals here. Whereas here, it's much lower, and here it will go high again. So, but then, okay. So that's the second thing, the third thing, if you like. And then what we can do is we can actually devise strategies of infection, if you like. So what we can do is we can say, okay, if the viral load, if the white blood cells, so the CD4, if that goes down, here you are in the AIDS phase, right? If that goes down, then for different regimes, if you like, of the healthiness of the individual, which is associated with the viral load, we can actually say, okay, there are different, we can identify different groups. We just say, okay, there's a group of 500, which is the CD4 expression 500, etc., all the way up to AIDS. And then we can say, okay, people might not know that they're infected. People might know that they're affected, and people might be treated, right? And not all that are treated, not all that know that are infected are treated. That's an important thing. But of course, all that are treated are know that they are infected. And so here you already see a start point of some behavior emerging. I hope you see that is. Because well, if I look at people that do not know that they are infected, they might behave different than people that do know that they are infected, right? So what we can do is we can look for all these 13 different populations. We can actually start to study how this whole process is actually happening, right? But before we do, in order to do that, we have to think about how the transmission of that virus goes through that sexual population in the case of unprotected sex. And so what you do here is basically you say, okay, if I have a virus somewhere in that network, and I take a snapshot of that network, what is the likelihood that that virus, given the network that I know, will be transmitted to another individual and just propagates through that network? Well, there you can write down equations for that. If you do that, you will find that it doesn't work. And the reason why it doesn't work is because you take too much a systematic approach. You're back again in the basic SIR models. I don't know if you know about these things. The basic partial differential equations. What you need to do is take into account behavior, right? You need to take into account the choices that people have, the choices that they have unprotected sex or not, the choices that they have in terms of their network structure. So that's what we're trying to do. And one of the things you can do then is, okay, how do people vary their relationship, right? So but then you get on, very, very difficult area because you have to start interviewing people and asking them. Now, we were very lucky. By the time we actually were at this point, we were very lucky because there was a new study coming out, the so-called Amsterdam cohort study, where homosexuals, MSM, were followed during 20 years. So during 20 years, they were interviewed almost every couple of weeks when they were getting new treatment. And they were interviewed about all kinds of behavioral, how their friends were, et cetera, et cetera, et cetera. And some of them were very open, some of them were lying. Of course, that's what you have at interviews. You know all about that. And so it makes it very hard, but nevertheless, you get data. This was 20 years of study of a significant population. And kind of things you can ask yourself then is, how do people change their relationship? So if someone is in a permanent, if someone says, I have a permanent relationship with this individual, this is my partner, what is the likelihood that he or she will still have a sexual contact, an unprotected sexual contact with another partner, right? So these kind of things you can actually measure, if you like, or deduce or infer from that data, and which is what we did. And I won't go through the details here because some of them are very gory, and other of them you don't want to know. But the bottom line is, you can actually, digging into these interviews, you can actually find interesting numbers as the probability of transmission. Here's something about this. This is an important thing. The steady partnership for homosexuals. This means, I'll get back to that number later, but that means that even if an individual says, in a homosexual MSM population, says that he has a steady partner, there is still 84% probability that he will have a sexual contact with another one unprotected within 1 year. I'll repeat that sentence. So if in an MSM relation, someone says he has a permanent partner, then there's an 84% probability that he will have a sexual contact with someone else in a year. These are numbers that we can use, and then we can do models, right? That's what we're all about. That's what it's all about, because we want to get our hands, we want to make sense of all these things. So here's just a very simple equation where you can do that. This says, the probability of transmission after an action, being unprotected sexual contact, between individual I and J is a function of some parameters. And one of the parameters, of course, is the treatment. So if the guy being treated, if he's treated, we know that the viral load goes down. If we know the treatment, we can calculate the binding affinity, we can calculate the viral load. Here you go, so you have the whole stuff. But also the statistical probability, which we know from literature. If there's a contact, and if someone has a fire, it doesn't mean that he gets infected. There's just a probability that it happens. And then this thing about the steady partnerships is an example, and the amount and the risky behavior. So that means basically not using condoms. And so, putting all these things into this complex network, we can actually propagate the virus through that network and see if we can retrodict what we know from literature, if you like. Now, I will show that result because I'm almost at the end of the story. I will show you that result, but before I do that, I want to come back to this point about controllability. And there is a nice paper that was written by Laszlo Barabasi and others, published in Nature under the title Taming Complexity. And so the idea was there that, the question was, can we control complex networks? And now what we all expected, and I hope that you expected, because I was priming, I just learned that word, I was priming your brain, that you expect that if I kick out the hubs, the whole network will collapse, right? So I did that on purpose, of course. And that's what everybody was thinking at that point in time still, in 2011, that by just taking out the hubs, you can actually, or manipulating the hubs if you like, you can actually manipulate the whole network. But it seems that it's not like that. And it seems that that it's not the hubs that drive the dynamics of the network, which is strange. I always thought it was like that. So it's very counterintuitive. They did that in a kind of analytical fashion, but they don't have an explanation. And I'm going to offer you an explanation for that, forever what it's worth. And the way we want to do that is, again, and this might be the red thread of my story, coming back to a basic entity rather than free energy, rather than entropy, well, maybe even entropy, which is information processing. So what we're going to do is look at the networks as an information processing unit, and then ask ourselves the question, can we direct that information? Can we predict that information, the way it goes through the network, depending on what we do with that network, which is controlling it, right? And so what the way we look at it is we have a network, so this is a slice of a network. There is an amount of information here. That can be viruses, that can be opinions, that can be anything, but I quantify it. My basic unit here is information. So my unit of complexity is information. And then I just look how that information propagates through the network. So this is a kind of symbolic representation of that. Now you have to figure out how to do that. And you can just put a package there and just send it around and hope that it arrives somewhere. But you can do it a little bit more structured, and that's what we did. So what we did basically is, for those of physicists in the room, we basically took an Ising-Spin model, or an Ising-Spin model if you like, and put that across the network. So what we did is, for those that are not physicists, what we did is we create such a network, and then we put on that network, that agent, if you like, a spin that can collapse. The spin is like a magnet. So magnet can be up, can be down north, south, or south, north, whatever you want, or can be down. And it influences other spins, right? So if I have a spin here that's up, and this one that doesn't know what to do, it can actually align. That's basically the idea. So what you can then do is, by using this concept of Ising spins, and I don't want to go into the details there, is actually calculate something like how much information about a certain part in the network is known and how that propagates through the network. So these are propagation functions, and how it stores in the network. So here you see a picture. This is the information in a node, which is depicted by this Ising spin that's flipping up and down. And then, depending on the connection in the network, that information can actually spread through the network. So we look at the dynamics of, if you like, correlation in the network, where we input a pulse of information and see how it spreads. You're still with me? Good. So that's what we're doing. And then if you do that, this is, and I want basically, let me say, well, let's look at this one. So if you do that, you can do that for different temperatures. That means the speed with which you fluctuate these spins. But if you do that, then you see that it actually goes to an optimum. And this, what you see here, is again the degree of connection. So the amount of connections that one node has, right? In terms of sexual network, the amount of sexual contacts that the individual has. And this is then the thing which you call information dissipation. That tells me something how much that information is spreading through that network. So what you see is that instead of what I thought, the ones with the highest connections will be the one that is spreading most of the information, but it's not happening. So the ones that really spread most of the information, relatively that is, are the ones that are kind of intermediate connected. And this might be a kind of an explanation to this paradox of Barabasi that these interconnected ones are more able to actually transmit information. And the argumentation about it goes like this. You say that the highly connected ones have stored all the information already in that network. If I take them out, their knowledge is still there, if you like. Their information is contained in the network. So, and then the ones that are in the peripheral, that are hardly connected, they are noisy. It doesn't matter what happened to them. They are so noisy they just disappear anyway. So it's again almost like, when I was in Santa Fe in '95, there was this whole thing about life at the edge of chaos, right? So complexity is what happens at the edge of chaos. It's a bit like this, right? Because if it's too noisy, the whole thing will not do anything. If there's no noise, the whole thing will not do anything. It's actually something in between. Okay, so then we put all the things together. I'm almost done. And so what we did is we actually took all these different simulations. So we took the molecular dynamic simulation, remember the calculation of the binding affinity. We took the immune system simulation. We took the transmission probability. We took the sexual networks. And all these parameters of these different models were filled with real patient data. And then we actually found this relation here. So this is reported data on AIDS cases from the 1990s up to somewhere in 2009. And the red one is the simulations, and the outcome of all these models that we have combined. And the blue ones are the data. So I was pretty happy with this, because it seems that we are able to retrodict the AIDS outbreak in the early 90s. And so this gives you confidence. So it gives you confidence in this tremendous complex thing where behavior and molecules basically talk to each other, and that you still can make some kind of sense. And then what you can do, and that's the whole reason why we want to do these things, the whole reason why we want to do complex systems research in the first place, is to do what-if questions, to run scenarios. If Peter Ho would be here, that's what he wants, right? He wants to run scenarios and say, okay, give me advice. What would happen if I do this? What would happen if I do that? And okay, so if you have some confidence that you're making sense, then you can start to play games and say, okay, what would happen if I change the scenario to more responsible behavior? Or I get better medicine? And hey, that's exactly the question we started out with, right? So we did that for a couple of big cities, Rome, Amsterdam, and San Francisco. And this is the Amsterdam case. Of course, the data get more noisy there because you have much less individuals. You would talk about 2,000 individuals. And here the red one again is the simulation, and the blue one is the data from the cohort. And we're actually predicting this upstream going on now, and that's happening. So there are new data coming out. So it seems that our predictions are working out well. So we're pretty happy with this, and it shows that you can actually do these kind of things. And then you can play around with the different scenarios, as I said, because that gives you some confidence that you can do predictions in the first place, which we published. I won't go into that. So the answer that we can give to the European Union is, when we played around with this, is what? What do you think? Why? Why?
Because it increases the transmission of resistance to the drugs.
Okay, so you say that that behavior increases the...
If the behavior doesn't change, the drugs become progressively less effective.
Okay, I never thought of that one. That's a good one. So you made a link between resistance and behavior. That's an interesting. I'll put it apart. I don't think it's the answer, but that's a very nice one that I want to think about. I need more time for that. So behavior is one. So who votes for behavior, and who votes for behavior? Okay, so that's about half. And let's say, I'm not sure it's half because there are always shy people never put up their hands. So I cannot do the statistics here. Okay, the story is that indeed it's behavior. But the reason is quite subtle. The reason is that if the medication gets better, and it's getting really, really, really good now, because HIV makes sure, I have to get this message across, HIV is not a death sentence. So the medications are getting very, very good. We have all nice combinations of cocktails that we can have very low density of molecules, if you like, and still working very well. So they get much better. So that means that with less medication, your viral load goes down faster, and so you feel better. So when you feel better, you might forget that you're ill. So if you feel better, you forget that you're ill, then your risky behavior increases. So what you get is a real trade-off between these two. So you feel better, you forget that you're ill, or you push it away, whatever that happens. I mean, it makes a difference if you stand up every morning and think, oh my God, how I'm going to live through this day, and or I have to eat these drugs again. Or you don't, you take a pill and that's it, right? It's a big, big difference. So the whole attitude with respect to the disease changes. And because of that, they might feel better, their risky behavior increases, and then the probability, of course, with respect to the contacts, increases again. But the viral load is lower. So, and if you do all these things, and if you look away at all these things, and that's what we did, then the conclusion is that eventually, if we would decrease the risky behavior with 30%, we would stop the pandemic. So that's a really spectacular result. So 30% reduction of the behavior. So that is in large cities. There's a caveat there. So in large cities, social policies by far outweigh medical improvements. But it's of course the combination, right? Because I said better medication. And so it's combination. It's really like, in physics we call it a frustrated system. You can turn here and then things happen there. So that's what's happening there. And we did more studies, which I have no time to go into. We actually combined that with models of test and treat. So if we would now go out and test everybody in the whole world and directly start putting them on treatment, then within a couple of years, the pandemic will be gone. But we don't have money for that. Anyway, so that's the kind of things you can do. So to summarize, this is my my overview slide, if you like. To summarize, what we have done is the following. There's one path that I didn't talk about. What we did is the following. We get patients in, right? We're actually using this system now. We get patients in. We measure their CD4. Remember, that's the telltale of the immune system, the activity of the immune system. We measure their viral load, right? And we look at the mutations, because these three we need. So the mutations, the viral load, and the immune system activity. And then we go to the left here. Then what we do is we calculate this protein structure, this binding affinity, so how good the drug will work for that specific patient with that specific set of mutations. This is really patient-specific. By the way, this is tremendously amount of calculations. We developed special techniques to use the supercomputers in the States and supercomputers in Europe to connect them together to actually calculate these things, but I didn't want to talk about that today. Then we do this immune system simulation, right? Again for that patient, so we know because we have his CD4, so we can do things there. Then we do this prediction of the immune system, and then we actually can say this would be the best drug for you, and how would that affect the population, in which we plug the population details. In the meantime, a thing that I didn't talk about, but an important thing is we do text mining. So in the meantime, we look into all the literature. There's 32 million papers on HIV-related diseases. So that's and documents, whatever you have there. So while I'm talking, and while I've been talking way too much, so while I'm talking, there is a program running in Amsterdam that's browsing the web for all kinds of information on the amount of, given this viral load, given this drug, what is the probability that the viral load will go down. And so that we gather also together, and then we give the whole thing to medical doctors, and then they can make a decision. So that's actually how all these things somehow work together. We have at this point in time, we have it running in 12 hospitals in Europe as a test case. We aim at the AIDS conference in December this year to release it worldwide. I hope we will make it. It's depending on many, many things. And there are a couple of patents to actually, safeguard our IP. Of course, there are many different, you were talking about where you get your funding from, so I have to do that also. So there are many different places where we get our funding from. Definitely I want to acknowledge NTU because they really take part in it. I think the complexity program is really helpful there. So my last slide then, if you want to know more, just type my name in Google, say I feel lucky, and then you might find me. Thank you for your attention.
Okay, thanks, Peter. Fascinating, as always. We have time for questions, so I can see one arm up already over there.
So, thanks for a really stimulating talk. I wanted to explain why I voted for behavior and see if you can see what your reaction is to it. If you look at the simplest epidemic models, most of the terms in those models are linear. The only one that's not is the transmission term, the nonlinear term. So it seems to me that affecting the transmission is much more likely to have knock-on effects, nonlinear effects, and reduce the spread. So
yeah,
that was basically my intuition.
Yeah, well, that's a very good intuition. But there is this connection between the nonlinear part and the linear part, right? Mm-hmm. So that makes the intuition, that it needs a bit more support for that intuition, so to say. Because you're right, the transmission part is more or less linear, more or less. It, depending a bit on the different type of sexual contacts, you have a slightly different behavior.
Well, it's bilinear.
Sorry?
It's bilinear. I mean, that's the quadratic term.
Yeah, no, okay, no, but okay, there are more aspects there. The drug-resistant virus has a different way of has a different transmission probability than the one that's not drug-resistant. So there are all kinds of lateral effects, so to say, that play a role. And then there's the behavior that comes in that actually feed back into that thing. So yeah, but you're absolutely right. That's a good point. But you have to take the whole thing into account, nevertheless. Yeah.
Professor Sloot, thank you very much for a really fascinating overview of your networking and modeling studies. You made a very fundamental assumption in the beginning, maybe not an assumption, but a statement that said that RNA transcriptase is a very error-prone enzyme, and it mutates, I mean, it has an error in transcription every time, right? And therefore, this underwrites almost all our efforts in AIDS drugs, because effectively you're saying that whatever molecule you devise, you will actually get the mutations which will then dislodge it, as you've shown in that modeling with the UCL colleague. So your data clearly shows also, with your correlations with population data, real patients, that behavior does dominate. Does it mean, therefore, that actually we may have overemphasized our resources in drug research? Because if your early statements of the mutations is a correct one, then really it's mutating every time. It's just like how we are attacking malaria and all. We are always fighting a moving goalpost. Yeah. So maybe we shouldn't. We just forget it. Let's attack.
No, be careful here. That's not good, because
yeah. So you made that statement earlier on.
I understand your point, but we really have to start thinking here as complex system scientists, right? So because everything we do will affect something else that we do, will then affect the things that we were doing. And so if we don't have optimal drugs, so to say, if we don't work on getting better drugs, then the viral load will be higher. And if the viral load will be higher and we have the same behavior, then the transmission of the virus through the population will increase dramatically, right? And that will then might or might not change our behavior, because then we see how, like in the situation where we had the pest in the Middle Ages, we actually changed our behavior. We didn't know what we were doing. We were changing our behavior because of what we were seeing. So you see this feedback loop that's happening there. So that's one remark. Second remark is that the reverse transcriptase, yeah, that's right. It does introduce these errors, these mutations, but it's not the only drug that we have. We have also drugs. I showed you all these inhibitors, and one of the drugs that's coming up now is actually the co-receptor blockage. It's tremendous. It's like almost like a detective story, but it was discovered in sex workers in Brazil that some of them are actually resistant for the HIV virus, and these were showing in the co-receptors a certain switch. So one of it's a technical detail. But anyway, so there are now drugs being developed to actually stimulate that co-receptor switch and to block. So there are many more ways to attack it. But the bottom line is, okay, I was giving this talk or part of this talk when we didn't have all these nice results yet, but I was giving that in Russia at some point in time for the Academy of Science. And then a lady stood up and said, well, we have a solution to this whole problem. Once we find someone who's infected, we just isolate him. That's another way of looking at it. And of course, it's nonsense, right? So what we need to do, and there's another thing, and that's we have to have this balance between what is happening in the population and what's happening for the individual. So if you say, we don't treat because then, we don't develop better medicine because then somehow the disease will die out, for instance, because people get so ill that they cannot interact with each other anymore. That's not the solution you want if you are a patient, right? So there's also this trade-off, what is good for the population and what's good for the individual. So you still have to develop medication that people feel good.
Yes.
And then they will change the behavior. That makes it difficult.
Yes.
Complex.
Can I conclude that then attacking reverse transcriptase is we should just veer off it, and go for other sites like co-receptors and other targets, because that's the conclusion that I draw from your statements.
Yeah, but yeah. So but these are more hard. These are harder. I would personally think that looking for inhibitors that avoid the virus to actually enter the cell is the thing that we want to do. Therefore, I very much would put my bets on this CRX4 co-receptor switch. But technically, it's easier to attack the reverse transcriptase because we very much know the molecular structure and the spherical hindrance in there.
Yeah. But it's changing all the time,
yeah,
so it's really not very effective.
Yeah.
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 Okay, there's one more question. Yeah.
Greg Fisher. If I understand this right, the EU asked the question: should we be putting money into sort of changing behaviour or drugs? And you concluded behaviour. But did you look at, this goes back to the question about control and influence, did you look at the extent to which, in general, governments can influence behaviour? Because I look at, I work in a think tank, and I take the view that governments on the whole historically have thought that they can influence behaviour more than they actually can. Okay, so, in fact, in some countries, governments are just so not legitimate that some people can actually do exactly the opposite of what the government wants them to do. So did you consider that when you
Okay.
sort of concluded?
Excellent remark. So two remarks there. The conclusion that we draw here, that you should devise policies to change behavior, should trickle down to the workers in the field. Should trickle down to the people, to the doctors that provide the prescriptions. And you have to realize that for this very complex disease, really very complex, you have to look at the genetic pattern and blah, whatever, the history of treatment, because the mutations, right? The patient that you get in on day one is a complete different patient that you get in 10 days later. You have to reassess the whole thing. And you know how much time they have per patient? 6 minutes? So what you could do as a government is actually somehow devise something that there's more time to actually, or that you can have a faster assessment. And these tools that I was talking about will have to give you that faster assessment. It's not going to work from the government level. If the government, writes somewhere on a web page, you have to change your behavior, that's not going to work. Although in some countries, like Singapore, things might happen. And actually, so there's a funny story. I don't know how much of it is true. And when this dengue was coming up at different clusters in Jurong Point, just next to here, there was this story that that they wanted to have that every individual would measure his temperature because that would be a telltale of what's going on with that epidemic, the dengue epidemics, and that there was a very high response. More than 70% of people actually did that. I imagine if I would do that in the Netherlands, I get shot. There's no way, but there's no way that it would indeed invoke an opposite reaction. People would just say, yeah, exactly. So yeah, they're all kind of, but so I was not saying that the policy change should come from the government, but the government should drive, should facilitate ways to change behavior. Yeah. We can take that away.
Kevin?
Just very brief
okay,
on that
okay.
one. I think it makes the point that your model shows where the leverage is. It doesn't say where the most effectiveness is.
Yeah.
Yeah.
You know,
Yeah.
This is about effectiveness of
Yeah,
policies. This shows where the leverage is.
Yeah.
Yeah. But you need these kind of models to actually, in the first place, convince the government that they can do something.
Yeah, one more question.
Yeah, okay. I noticed that you mentioned Barbasi's paper on the contributivity of the network. Actually, shortly after his paper was published, there was another paper published in PROS1. The author actually, I think the author communicated with the Barbasi's group. So their argument is that Barbasi's analysis is totally correct. But it's totally correct, 100% right, no problem.
Correct, okay, I thought yes. You said crap.
Sorry. Okay, but their argument is like, basically, but they make an assumption on a network model, which may not be so realistic. Basically, in their network model, basically, it's like on each node, there is no self-loop. Right. Meaning like the node's next moment status basically only decided by the neighborhood status and their input.
Yeah.
They argue like in the real life, most of the network actually is not like that. It's like what we are going to be tomorrow is quite strongly related to what we are today.
Yeah.
So if you put in a self-loop, you still use the analysis, you will find that the hubs, those high-degree nodes, they come back. Yeah. You have a control over those several nodes. You have a control over the whole network. However, meanwhile, I found that this conclusion doesn't, in my understanding, doesn't really affect your study on the transmission, effectiveness of those low-degree nodes. So I'm just thinking that maybe the control of the network and the transmission of the information in the network, they are probably still two different things. That's my feeling.
Okay, I wouldn't know. I wouldn't know because I didn't look at that at that experiment that these guys did. So I wouldn't know. What I do know is that we applied this whole concept to something completely different, which I had no time to talk about, unfortunately, because it would have been fun. That was about criminal networks. So we have lots of data coming from the Netherlands. We have lots of data on drug use. And so we have lots of data on criminal networks. And one of the things that we did is analyze the criminal networks and then ask ourselves the question, what would intervening in such a network amount to if you want to stop the production of cannabis, for instance? So then you come to the same kind of question because you want to control it. Do you take out the one that is connected to all the other ones, the one that produces the plants, the one that harvests, the one that takes care of the electricity, etc.? So do you take out the hub, or do you take out the in-betweeners, the one that has the highest in-betweeners, or the one with a specific function? And so it's almost 10 times more complicated than we think. It's not, of course, only the topology. The topology is just one aspect of it. It's also the functionality of the individuals in the network. And the topology is changing over time. So I think the answer, you have to really look at each network separately. And then I think this information propagation can help you to actually assess where is the hot spot or where is the sensitive spot in the network. But you have to do that for each realistic network time and again.
Okay, I don't know, Jan, we're supposed to adjourn at 5 p.m., but I guess we don't have to rush or leave the room, or? Okay, so if there's more questions, we still have time.
People are eager for coffee now, I think.
Yeah, coffee or, okay, right at the back. You take the microphone.
Thank you, Philippe Michael from Division of Physics. Thank you for your talk. It was very interesting again, as usual. I was very curious about the diagrams which you built, which are representing the different population, right? So heterosexual, homosexual, and drug users. I wanted to just to ask a little bit about drug users. Is it both homosexual and heterosexual together? And I was also very interested why it's so separated. Is it just because of metrics, or is it seriously so separated from both groups?
Okay, so the drug users were homosexuals, heterosexuals. It doesn't matter. But once they were drug users, we disconnected them from the other clusters.
No, because
just to study the structure by itself, and then so we took them out from the homosexual population and the heterosexual population.
I see. So because it seems very, very strange that they have no connection.
Of course, there are connections. Sure.
Yeah.
But because we wanted to understand the structure of that separate network, the subnetwork, it's a subnet. There are networks that drive the dynamics actually. So we want to take out that structure, and the connections are actually very, very poor, because the size of the ones that are both IDUs, so drug use infected, and homosexuals is very small. So the overlap is pretty small.
And also a small question about this diagram. It was built for some particular city, right?
Yeah.
And how does it differ from all the cities which you study? I mean, was it some crucial difference, for example, from US to Europe, or is it always pretty much look like the same?
So what you were looking at was actually a combination of the three cities I was talking about: San Francisco, Rome, and Amsterdam. So there was an aggregation of these three cities. Between the cities, the differences were not that big. No. So also the gamma of the three populations are almost equivalent within the error bounds.
And through the time, also the evolution of this.
That's another good question. So we have time points because we have like over 20 years, like I was saying. So we can look at different points in time. But the difficulty is that when you go further in time, you get more data. It's more rich, and the errors are smaller. So how do I correctly compare these different longitudinal moments? It's very hard. But what you see is you see actually that the gamma starts to kind of how do you say that, asymptotically go to a certain value over time. So it seems that it's a stable factor. Yeah.
Thank you very much.
Yeah.
Okay, any more? Yep. You'll never get a chance to leave.
That's okay. Hotel California.
Vaishak from Computer Science and NTU itself. So I was wondering, you just said that the cities had similar networks. They were Amsterdam, Rome, and San Francisco, but wouldn't it be very different in the countries where AIDS is most prevalent, in Africa and Asia and all? The culture is completely different, right?
Yeah.
These networks would have a very different structure, I'm assuming, and would the same spreading effects and all be the same?
Yeah, so I was waiting for that question. Thank you. I didn't instruct you to ask it, though. No, but that's a very, very, very important point. How much of what we have done can actually be translated to countries like African countries? So we have collaboration projects now going on. We just started it like 3 years ago with some Zimbabwe and Kenya, I guess you say, where we're actually trying to do the same thing. The point is the data is very poor there. So we're now setting up posts, that's done together with Erasmus Medical Center. We're now setting up posts in these countries to actually get that kind of data. That is very, very poor. And also the cultural differences are very much different, of course, than we have in Europe. That's one thing we're doing. Another thing we're doing is in the slums in, you know of that, in the slums in Bangalore, there are 36 slums. So we're getting data from that now. That's over the past, that's 3 data points where we get all this kind of information. I'm very much looking forward to mining that data to see what's going to happen there. So it's data for, and that's, again, because it's a completely different culture, you might have a completely different effects there. So we will look into that. Sure.
Okay, any more questions? Oh, well, okay, 2 more. Yes.
Okay. I'm sorry, I'm not sure whether this question was asked before, but you say that noise helps to transmit information within your network.
Could you say it again? I said that?
Noise.
Yeah, yeah, it's good.
Noise helps to transmit information in your network.
It helps, yeah.
Yeah, could help, but how much noise should be added into your network so that you really maximize the efficiency of the transmission process?
Yeah, that I don't know. That depends on the network. So we did that for 5 different artificial networks. It's in the paper, you can actually look it up. 5 different artificial networks which you can generate, you just have different gammas. And then we found that, you know, up to something like, even sometimes up to 20% of noise will actually still enhance the signal-noise relation, if you like, of transmission. So that's pretty good. And then we did it for this email network that I was talking about as a realistic network. So we tested the gammas that are close to the homosexual and heterosexual populations, and we took this hierarchical, which is a slightly different type of network of the email network. And for the email network, it was pretty small. I don't remember, it was like 5% or so. So you can add 5% noise, then the transmission of information increases. You go beyond 5%, then it decreases.
But should it depend on the topology or the functionality of different nodes when you're adding noise? I mean,
this was homogeneous in that sense. So the network topology we changed because of this gamma, but the activity of the individuals in the nodes, so the agents, was all the same. So they had the same rules there. Yeah. If you changed it, of course, if you changed it, that might also have an influence.
Okay, I think in the interest of time, we should probably leave the remaining questions for reception tonight, which is at 7 p.m. 6 p.m. 6 p.m. So the last thing to do, just for me to thank Peter with one hand.
Okay. Thank you.